Hey everybody, we're just gonna make our video and go through the questions on how to do them. So the first one, it says, suppose a eukaryotic messenger RNA has the following nucleotide sequence. It gives the sequence. It says, assume the RNA polymerase to produce this molecule from right to left. So we know if it went from right to left, we know the right side is going to be the 3' side, because it started on the right and moved to the left, and it always synthesizes strand 5' to 3'. So the right side of it has to be 5'. So that would mean that it'd go 3' dash UGCUU and carry on, and then AGA 5'. And then it says for part B, it says what is the sequence of the template strand. So the template strand is always complementary to the RNA mRNA molecule. So basically you just line up the 3' to 5' in the exact same order and then list it out. So it would be like 5' ACGAA CGG TTT AGT AGG CT ATCT dash 3'. And the next one is the coding sequence. So it's going to be the exact opposite of the template. Just remember, you have to flip it and make it 5' to 3' to make sure it's always, when you're synthesizing, 5' on top, 3' on bottom, and they go across. So that works. For the picture, we have it right here. So hopefully you can see it. So you're going to have your coding strand on top, which starts 5' and goes to 3'. Probably looks backwards on this. And then the template strand goes 3' to 5'. And then the DNA polymerase, that's just our little box, even though it really covers both strands, just to show where it's synthesizing exactly. And then that goes 5' to 3'. So you started it started synthesizing the 5' end, and then it's going to work its way to the 3' end. That's how you do number 2.
All right, so number 2, it's just to fill in the blanks. A says that the blank binds directly to the TATA box, while blank binds directly to the -10 and -35 sequences. So the first blank on that is going to be the TBP, which directly bonds to the TATA box, and the sigma factor is what binds to the -10 and the -35. For B, guanyl transferase is a eukaryotic enzyme that links the 5' carbon of one of the nucleotide to the 5' carbon of another nucleotide. So that's the guanyl transferase. C is group 1 introns is the first blank. Those are the things that use nucleoside guanosine as a cofactor during catalyst, while group 2 introns use adenine as a cofactor during catalyst. D is the CPSF, is the endonuclease that assists in terminating transcription in eukaryotes. E says that in lesson 9 we learned about general transcription factors. So TF IIB is a GTF, which acts as a bridge subunit to link TFIID to TFIIF/RNA polymerase 2. And the second blank is TFIIH is a GTF, which acts to phosphorylate and activate RNA polymerase 2. For F, the spliceosome is composed of subunits called SNRNPs. These spliceosome subunits are composed of 2 components, proteins and smaller nuclear RNAs. That's number 2. And then number 3 says, this question concerns the proteins involved in transcription and RNA modification. So going through the different proteins, A says indicate 2 proteins that are either prokaryotic or eukaryotic that act as helicase to separate 2 DNA strands during transcription. TF, RH, and RNA polymerase 2 are 2 proteins that do that. B asks for to indicate 1 protein in prokaryotes that act as helicase to separate RNA from DNA during transcription. That is the Rho protein. C says indicate 3 molecules in eukaryotic that function during RNA modification as ribosomes. The SNRNPs do that, as well as group 1 and group 2 introns. For D, it says indicate one protein in prokaryotes that act as a transcription factor, and that is the sigma factor. And E says indicate one enzyme in eukaryotes that synthesize a long string of identical RNA nucleotides, but does not use a template DNA strand for RNA synthesis, and that is poly-A polymerase.
Okay, so for number four is a little difficult. Our drawing is fairly poor and somewhat incorrect. So we're just going to kind of walk you through how you should do it. So what you need to do is you need to draw your template strand, which is going to be 5' to 3', and then your coding strand, which is 3' to 5'. Yes, that is correct. And then you're going to put your coding strand is going to have an R loop. So it's going to have a big R loop kind of like this. I can show on my iPad. And then this is a different color. And then here's this. So it's going to be like that. If purple was your coding, black was your template, and then red was your RNA. And make sure this is 5' to 3', 3' to 5', and then 3' to 5'. Like this. So you're going to make sure that happens. And then for the next thing that'll happen, for the next question, how it says the mature mRNA for YFG is hybridized to the genomic DNA copy. You have to remember that the RNA doesn't make the loops. It's going to be the two strands that make the loops, and especially the coding strand. So the top strand is going to make the R loops. So these are all R. And then the template strand is going to make the introns. So those are where the introns are located. Let's say if they're blue, they're going to be located like right in here, right in here, right in here, and right in here. So those would loop out like this. And then this strand would be changed to look something like this. A little R loop and then a big loop, a little R loop, a little R loop and a big loop. So horrible drawing, or I guess they're not really even like that. They're supposed to be all looped together, but you get the point. And then a loop, and a loop. And the loops between the introns, that's where the R loop is for the mature. And then to go to the next one, it says the mature RNA for YFG is hybridized to the genomic DNA copy of its YFG. In this case, the 5' most intron of the pre-mRNA contains a defective donor site and branch site adenine. Assume that the introns are removed and used in the spliceosome method. So what that would happen was this intron right here would get removed and would cause that this whole thing to become an R loop. Oops, I just deleted that. Become an R group because that intron, the spliceosome spliced it out. And that's going to be very similar for the next one, but it says the second and fourth. So instead of that last one, it'd be the second and the fourth one, which depending on, I don't know now, but depending on which one, it'd be the second and fourth going from the 5' to 3' direction. So it would have been this one and this one that got deleted out, and these two would have stayed. And that's kind of how you do it. It's kind of confusing, but that's kind of what happens. And then for number 5, it says there's a big list of proteins or sequences or things that are involved in transcription. And it says to list them. So like A is which of the above is found in bacteria, in E. coli cells. And so for a prokaryotic cell, E. coli, it's going to have the -35 RUT and NUSA with those. So that's kind of what you just do. You just figure out which one of these bullet points goes with the question. So the next one, which one above is found in yeast. So eukaryotic, and it's going to be the -25 enhancer silencers, group 1, GC box, TFID, and SNRP proteins. The next one, C, is which one above is involved in transcription initiation. So initiations where RNA polymerase 1 binds. So what things involve that? It's going to be the -35, -25 silencer, enhancer, GC box, and TFID. And then for termination, so the two proteins or the two sequences that are involved in termination are RUT and NUSA. which is above is a DNA sequence. So which of these ones are actual nucleotide sequences? And that's the -35, -25 enhancer silencers, and then the GC box. And that is how you do the homework. Have a good week, good Thanksgiving. I hope we don't have stuff due for Thanksgiving. Have fun on the test.